Good afternoon, everybody, or I should say good morning to those of you that are in time zones that are still morning. I'm Elaine Lyon. I'm an associate professor at the University of Utah and a medical director at ARUP Laboratories, and I'm welcoming you to this web conference. I wanted to remind people that they can submit questions anytime during the presentation. I may not be able to get to them right then. I will try to monitor it some in case I can answer a quick question at that point, but if not, I will answer them towards the end, so please submit your questions. I will be talking today about pharmacogenomics, next-generation sequencing, and clinical interpretation. I work in a clinical CLIA reference laboratory, and we have been working on next-generation sequencing and have several next-generation sequencing assays available, but we don't have pharmacogenomics assays available yet by NextGen. So what I'm presenting on today is kind of a proof of concept or a proof of principle, and the work that we're doing to evaluate whether we can use the next-generation sequencing for our pharmacogenomics. The disclaimer I will have on this is that this data is very complex, and so this is a very early step into it. We have a lot more work to do before it really becomes a clinical test. But I wanted to show a couple of approaches that we've looked into that may be suitable for the clinical laboratory. I do have a disclosure. I'm on an advisory board, although there's not a direct conflict of interest with what I'm presenting today. So I wanted to talk today, I'll give somewhat of a background of the pharmacogenetics and then go into some of the targets we'll be looking at. I'll then go into next-generation sequencing and exome data as well as gene panel data. And throughout the time, I am going to focus on interpretation and some of the challenges that we have with pharmacogenetics that are even above normal challenges of next-generation sequencing. So I'll start with the definitions. And you hear a lot of the terms personalized medicine or pharmacogenetics, pharmacogenomics, and I think those two are being used interchangeably, although pharmacogenetics should refer to a gene, whereas pharmacogenomics should refer to multiple genes or the genome. And then companion diagnostics. So personalized medicine is a systematic use of information about an individual to select or optimize preventative or therapeutic care. Pharmacogenetics then examines the impact of genetic variation on the response to medications. So this is to tailor drug therapy at a dosage that is most appropriate for the individual patient. So sometimes personalized medicine and pharmacogenetics are used somewhat interchangeably. And then companion diagnostics is a portion of pharmacogenetics. And these are assays that are used for a specific therapy to stratify the disease status or select the proper medication. So it's a bit more of a specialty section of pharmacogenetics or personalized medicine. And I will be talking about a little bit of some of these today. So there has been a lot of talk about personalized medicine or companion diagnostics and the promise that this has. And so when we talk about companion diagnostics per se, that's usually a single assay and it's done with a specific therapeutic product. So the concept is to identify patients most likely to benefit from a therapy or those likely not to respond or those likely to have an adverse reaction. These types of tests are considered high risk by the FDA, and the reason is that if you give wrong result, it could lead to withholding appropriate therapy or administering inappropriate therapy. So there has been some criticism that the genomic medicine hasn't realized the promise for personalized medicine. And it's true, we still have a long ways to go in understanding the genome, but there are some success stories, and in some ways that we are using and that we have realized some of the opportunities of personalized medicine. In the oncology field, looking at somatic mutations, there has been advances in tumor biology, understanding the tumor biology, understanding the expression of genes, and then also understand mutations that may then make you resistant for a certain treatment, etc. In inherited or germline variation, we talk mainly about enzymes, and so the cytochrome P450s, the UGTs, but we also have to realize that there are also receptors that are very much involved. And in an emerging field is looking at host factors that predict treatment for infectious disease. Now that's different than infectious diseases where you're looking at the viral genomes and the resistance mutation. And I noticed a mistake in these slides just last night. The IL-28B and the HLA-5701 are examples of the host factors predicted for treatment in infectious diseases. So those two need to be put up in the previous bullet. But the laboratory community has embraced, in areas that do have clinical utility at the moment, some of the personalized medicine and pharmacogenetic testing. Now, I will be focusing on inherited or germline variation today, so mainly the enzymes and that are involved with drug metabolism. So when we talk about drug therapy, we talk about pharmacokinetics, or in other words, what the body does to the drug, which are the metabolizing enzymes. But please also keep in mind that there's the pharmacodynamics, meaning what the drug does to the body, the response, the mechanisms of actions for these drugs. Now, the pharmacokinetics, when they looked at a dose response and the dose required for a desirable response, in the little chart at the bottom of the slide, it was noticed that they were grouping into approximately 3 different groups. And this actually then could be related to the drug clearance, and actually has been shown to be associated with genotypes of specific genes. So for example, if you have a poor metabolizer, or a slow metabolizer, you will have non-functional genes, or a fast metabolizer, and the genotypes on the top show you the different types of genotypes. So a slow metabolizer would have both of the person's genes knocked out or basically non-functional, whereas an intermediate metabolizer would have only one of the genes non-functional, a regular normal 2 functional genes, but then you have a fast metabolizer that could actually have duplication of genes. And this is for my my pharmacology friends, to just point out that it's not all about DNA. So genes are important, but in the pharmacology field, the formulation, the body mass, the age, concurrent medications, and even diet all affect how drugs work for an individual. And also for my pharmacology friends, that there are limitations to the pharmacogenetics tests. And I think this is one of the take-home messages that I'd like you to have. So first of all, genotype, does it always equal phenotype? It makes the interpretation of results difficult if that's not always the case. It also makes the clinical application of the data difficult. One of the reasons for this is that the pathways may be necessary to understand, and looking for different variants in different genes of the pathway. So if one gene product is not being produced, it could be another pathway is available and could use that. So we do need to start thinking in pathways and not simply individual genes. So the content of our pharmacogenetic tests varies among laboratories, even for the ones that are existing. So one reason is that there are licensing issues that are still in play. Even though there was a Supreme Court decision about DNA as a naturally occurring substance, there are still a lot of questions about the licensing. then the question is, there are clinically significant single nucleotide variants, but then there's an awful lot of variants that are not well characterized. They may be called polymorphisms, but we really don't know what they do. So we still are struggling with understanding which are the clinically significant variants. Gene dose is very important. As I showed before, you could have duplications of genes. You can also have deletions of genes. And in many cases, or in some cases, the alleles are described by a combination of variants on the haplotype. And right now, all of the genetic tests that's being done is genotyping. We are not doing molecular haplotyping. So we need to infer the haplotypes. And we do that based on known linkage between the variants. Now, one of the challenges with pharmacogenetics is that molecular testing isn't necessarily a very quick test to do. It's not quite as simple and isn't typically done in stat labs or in a stat time. So for example, with us in a reference laboratory, it would take a day for the sample to get to us. Through our system, we need to then extract the DNA from the sample, run the test, and some of the tests may take several hours. When you talk into next generation sequencing tests, actual running the test takes days. So this is not going to be a quick turnaround time, and yet in many cases that's exactly what is needed. If somebody comes into the doctor, they need to get started on a medication quickly. There isn't a time to do the workup before they may need to try the medication. And then the big question is the pharmacoeconomics. Can we, using our pharmacogenetic tests, be able to really make an impact on the patient care and the costs of healthcare? Okay, so now that I've given you all of the limitations for pharmacogenetics tests, I want to go into a little bit of the background of some of the terminology. So we classify them in different categories. So an extensive metabolizer is actually the normal metabolizer. A poor metabolizer means that you have no functional protein. On the other hand, an intermediate means you have some functional protein, you have some enzyme activity, but not the normal number. And then obviously the ultra-rapid metabolizer, which would be the multiple copies of functional genes, or the variant in regulatory regions that increases the expression. Now, as I said, we put them into categories, but please realize that this is more of a continuum. We don't exactly know. An intermediate metabolizer may actually be 50% of normal, but it may go down into the 20% of normal or even below. So I also like to think of this as a continuum and not simply 4 discrete categories. Now, to make matters more complex is that the enzymes are often considered recessive patterns. I'm not going to say it's a recessive disease because none of these cause diseases. You don't even know about them until you are challenged with an exogenous substance such as a drug. But it does show a recessive pattern in some ways, that to be a poor metabolizer you have to have 2 copies, one on each allele of non-functional ones. So this slide simply shows the different combinations. 2 functional alleles will be extensive. A functional and a decreased functional allele will still be considered an extensive metabolizer. A functional and a non-functional allele combination will give you an intermediate, or some people still also call that an extensive metabolizer. So there's some discrepancy between what different laboratories may call the functional non-functional genotype. A decreased and non-functional alleles would be then intermediate. 2 non-functional alleles would be poor. A duplication and then functional alleles on both will be ultra-fast. But there's also known duplications of non-functional alleles. So it is important to know if the duplication is a functional or a non-functional allele. So how are the data used to select or avoid the drugs? And we need to also think in two different ways. So one is that some drugs are inactivated by the pharmacogenetic gene product. So if they are a poor metabolizer with a drug that's inactivated with it, you want to avoid the drug or use lower doses. The intermediate metabolizers also may require lower doses, but the intermediate categories have been less studied, and there's simply less good data for that. The ultra metabolizers may require a higher dose, or they may not respond at all to the drug, and you may want to avoid it. There are some dosing guidelines that are published, and again, this does not replace the need for therapeutic drug monitoring or other clinical monitoring. Now, on the other hand, drugs may be activated by a pharmacogenetic gene, so it may be a prodrug that only then becomes activated with a metabolism. So in this case, if you're a poor metabolizer, then again, you may want to avoid the drug or give a higher dose. The intermediates may require higher doses. The ultra-fast metabolizers may require lower doses. So when laboratories try to do an interpretation, it really depends on the drug that they're given and whether it's a prodrug that's activated by the gene product or if it's a drug that's inactivated. So the common genes that we look at, and then go into these genes and others with the exome and genome sequencing. So cytochrome P450 2D6 is a enzyme that is responsible for the metabolism of nearly a quarter of all drugs. And it's also commonly involved with drug-drug interactions. The genotypes are given star allele numbers, and so this is a challenge with our standard nomenclature in molecular genetics. And as I commented before, that the haplotypes, the combination of variants on one allele, may be how these alleles are described. The phenotypes, 60% are extensive metabolizers, but about 10% of the people are poor metabolizers. And then others, intermediate or ultra-fast. Here are some of the common CYP2C19 alleles, and I'm showing this to you so you can see the number of nucleotides. So for example, the first one is star 2, and there'd be 2A, B, or D. There are some minor variations between those in what variants are on the allele. But 2A has the -1584G. That's a promoter variant that can affect the expression of the gene. The other three are fairly common variants. A star 3 is a deletion of an A. A star 4, you can see, has 100 plus 1661 is a common variant. 1846 is what we would call the allele-defining variant. That's the mutation that causes it to be non-functional. In this case, it's a splice site variant. Star 5 is the entire gene that's deleted. And then I wanted to skip down a little bit to star 10. So if you look at star 10, it has the 100 C to T and then two common variants. If you get a 100 C to T and an 1846, those are assumed to be together, so that would be a star 4. Whereas if you see a 100 C to T by itself, it would only be the star 10. The star 10 has reduced activity, whereas the star 4 has no enzyme activity. So this becomes a little bit challenging if clinicians are not understanding how laboratories are assigning the phenotypes. A star 4, star 10 compound heterozygote would be seeing one copy of 1846 and then two copies of the 100C to T. So it does become somewhat complex in understanding what the haplotypes are. Now, I also wanted to comment about cytochrome P452C19. In this case, there are two common variants that predict poor function, and very common in the Asian population, less common in Caucasian or African Americans. There's other known variants, which also includes a star 17, which is a rapid metabolizer phenotype. So it actually increases the expression of the gene. This has got some notoriety recently because of clopidogrel, which is a platelet aggregator. And it is considered a prodrug, and it's activated by a number of different cytochromes, but including CYP2C19. And there have been a number of studies showing that failure to respond is associated with genetic variants in the CYP2C19. There is always the challenge of deciding whether you go with a genetic assay or a functional assay, and in this case a platelet functional assay. And each of them have their pros and cons, but one of the advantages with the genetic assays is that you actually could identify it before a person is actually put on the drug. Then the one last category I wanted to talk about is the host genetics and infectious diseases. So there are two examples, hepatitis C with interferon and IL-28B. Now we all call it IL-28B, but it really isn't IL-28B. They are regions close to it. It's the closest gene with it. There is an assumption that it probably is something within the IL-28B, but it has not been identified yet. So these variants in the IL-28B region is associated with spontaneous or treatment-induced recovery from HCV infection. There's also an example for HIV and abacavir therapy with the HLA-B5701 allele. So the hypersensitivity to abacavir is strongly associated with this allele. It has a very high negative and positive predictive value. So by testing for HLA-B5701, you can reduce the incidence of a hypersensitivity reaction. And this is recommended by the FDA for pre-therapy screening. Okay, now I want to switch a little bit, switch now to our genomic sequencing assays. And in genomic sequencing, we do multi-genes, exomes, or genome sequencing. Each of them have their advantages and some disadvantages. So with multi-gene panels, we do only known genes associated with a specific disorder. We also look at intron-exon boundaries. It has a very high depth of coverage, but again, it's limited to just that known set of genes. With exome sequencing, when we say exome, it's not 100% of the exome, but we can get about up to maybe 95% of the exome. This again will be all known genes in the exome and the intron-exon boundaries. There is an intermediate depth of coverage between a gene panel and a genome, and when you start analyzing at an exome level, there is a challenge in the interpretation. Genome sequencing essentially covers all of the exome. It does include genes, the inter and intragenic regions as well. It has a lower depth of coverage, but a very high breadth of coverage. To handle a genome, it's first analyzed similar to an exome. And then if causative variants or mutations cannot be identified, then we'll go to doing additional data and reanalyze. So I'm going to start with the clinical exome, or the genome. And the clinical exome is available in clinical laboratories, and one of my colleagues will be speaking about it this afternoon. But when we analyze the clinical exome, again, we start with genes associated with disease. So do we limit it to genes in the online Mendelian inheritance in man or the human genome mutation database? Well, that's where we definitely need to start with. And then when we do an exome, we will find incidental findings that are not related to the disease at hand. There is a recommendation from ACMG for actionable. And this is where I put in our pharmacogenetic variants, that it also could be an incident. I wanted to also point out that in the clinical realm, we don't do gene discovery really when we're looking at the clinical exome. That is in the R&D at the moment. But with that said, there is somewhat of a precedent. When we went to Sanger sequencing, all of a sudden, even though we knew the gene was associated with the disease, we were doing mutation discovery. So for a number of years now, we've been doing mutation discovery in the clinical laboratory. So are we approaching a time when we actually could do gene discovery in a clinical setting? In analyzing an exome or a genome, we take advantage of filtering, and we can do this because we're looking for rare variants. These, the diseases that come to us for exome analysis are ones that they're unable to find the cause for the disease. They are most likely very rare diseases, so we use symptoms to guide our analysis. That means that we can filter out common variants. So we filter out known benign variants. We use dbSNP and the common SNPs, or the 1000 genome, or the SP5400. So these are population, basically population sequences. And what's even more important is that we use the frequency data. So for example, if something is found in 20% of the population, a variant is at 20% of the population, but the disease is a very rare disease, it's fairly safe to say that that variant is not causing the disease. So again, as we're looking for rare variants, we will not start with intergenic regions. We'll probably filter out even deep intronic regions for a first go around. We will look for variants of interest, meaning non-synonymous variants or those known to be deleterious. And I will just put a plug in here that we all need a better clinical database. And NCBI is working on that with ClinVar. And hopefully that'll be a curated database, and clinical laboratories have agreed to be submitting information. Again, we can look at regions of interest. So even though we do an entire exome or genome, we can really narrow it down. We can look for candidate genes, or we can look by chromosome, or we can look by variant. And so now I wanted to talk with these incidental findings. The American College of Medical Genetics, they list 56 genes. These are medically significant, and they are actionable. In other words, there are treatments available or some other way that it will alter management of the patient. And when we are looking for these, we need to do a second analysis specifically for these variants, because we may not find them by our symptom-guided analysis when we're really focusing on the disease and the symptoms in trying to determine which genes and variants may be causative. So what about the pharmacogenetic results? Again, these are germline variants. Can they be included similar to an incidental findings? In a sense, they are considered actionable if a person is going to be going on medication. It may be good to know this. So is this something that we can add to our exome and give pharmacogenetic results as a secondary type finding to our exome findings? There are discussions such as what type of informed consent or approval would we need from the patient. And these are being worked out for the ACM gist of incidental variants, but has not been talked much in the pharmacogenetic setting. So looking at the pharmacogenetics from the exome genome, here again is my disclaimer of all the limitations and challenges. First of all, it's not disease associated, and so we can't use OMIM or HGMD to look for genes with specific symptoms. And since they are common variants, often we can't filter by population frequency. So two of our best ways of analyzing variants for rare diseases are not available to us for pharmacogenetics. We still have the challenges of variants outside the region or intron-exon boundaries that we would need to design around, and repetitive or GC-rich regions are going to continue to be a problem, as they are with all genes. So exon 1 often may not be covered very well. One of the main problems with doing pharmacogenetics is that there are many of these genes that have pseudogenes involved with them, or what are called segmental duplications. The copy number variants are not well technically validated yet by next generation sequencing. We hope that we can, but not necessarily. We may need to have some workarounds. Again, we'll have to have workarounds for structural variants such as translocations, inversions, gene conversions. Now, one of the other challenges from the exome or the genome is that we need to translate the star alleles or the haplotypes into RS numbers or to standard nomenclature. And this actually has been a challenge, and I will show you some of the challenges we have of just making sure we're looking in the right position. And then, as always, there are some uncharacterized variants. This just shows an example of the list when I first tried to pull out some genetic variants from an exome. And we would get a very long list, but sure enough, I saw a CYP2C19 variant, and not surprising. And the analysis program that we have can give us a lot of information, and it can pull up the gene and let us read about the gene and look at some of the phenotypes associated with it. We can look at our own internal frequency. We can pull up frequencies from other places. It'll give us an RS number and references with it. So as we go through this, we can then go variant by variant and see if it's going to be something that would be significant. Now, that is very challenging doing that with an exome. So even though we have all of that data, we may need to narrow it down. So when I first looked at an exome for pharmacogenetics, I could pick out a couple of CYP2C19, etc. But it was very challenging going through all the variants that we pick up with an exome. So our next step was to say, okay, even though we have an exome, we don't need to analyze it as an exome. Let's analyze it as a gene panel. And so we chose the known genes. These include the CYPs, the SALTs, UGTs, different ones, and identified which genes we wanted to look at. And so now we're not doing a whole exome. Even though the data is for the exome, we're now narrowing it down to only known genes. Now, again, we went back and looked at all of these genes and said, how many of them are we going to have problems with because of pseudogenes or duplications? And as we looked at that, there were a number of genes that are affected, such as CYP2B6, 2D6, 3A4, 3F2. The HLA-B, there's a lot of variation within that region. IL-28B, and realize that we're not looking in the IL-28B gene. We need to go custom design the regions associated with those variants. UGT I thought was very interesting because UGT 1A1, 1A3, 1A4, A6, and A9, they all differ only by the first exon. So it may be difficult to determine which gene really has the variant in it. So I took our exome samples and I wanted to look at we had 14 different exomes that we looked at, And I could not filter by common variants, by rare variants, so I had to keep the common variants. I could filter by deleterious effects for things that are known deleterious. I could filter by quality scores, so to make sure I'm getting real variants and not genomic sequencing artifacts. But again, I had no disease to associate it with. And I took just one gene, and we really need to do this with all of the genes, to look to see what the coverage is for this gene. This is DPYD, involved with 5FU. And as you can see, we get variable coverage throughout the exomes. And so to really understand what is coverage, we need to get down into the exome level on a gene-by-gene basis. Here is a summary of these 14 exome samples with total variants, then single nucleotide variants. We're given a quality number and a transition-transversion ratio number, but if you look at all of these, I had approximately 100 to 140 variants per patient that we would need to look through very carefully to see if there was anything there. So when I then filtered and used some other filters with this, I was able to pick up and narrow it down to 57 variants. And so this one example I wanted to use is that we picked up a variant in CYP4F2. Now, first of all, we know that that's one that has a segmental duplication. So we need to figure out if we're looking at the real gene or if the segmental duplication, and it may require a handling this separately. Now, it was a missense mutation, so it wasn't an obvious pathogenic mutation. It had a very low frequency reported in the population. So again, I don't know how to think about it at that level. So obviously not much is known about it. It was a very low depth of coverage, and so I have here, and you can see that I've only picked it up maybe 6 times. Is that real even? So we would need to Sanger sequence it to confirm even if it is real. And then the other difficulty is that it's not even listed in the allele nomenclature, the database that we use for CYP4F2. And so again, this would then become an uncertain variant that we wouldn't know how to handle clinically. Then when I looked at this sample and pulled up, it had a lot of HLA variants with it. Now, we really are only interested in a couple of HLA variants, such as the 5701. So maybe this is when we started thinking, maybe this isn't the approach to take. Maybe our approach is that we need to start analyzing by known variants. So this is a chart of the 15 different samples that we used, the 14 different samples with the known variants, things we already know how to interpret, and that we can look to see what their genotype is. So if you can look around here, there are a couple of CYP2C19 variants that we are picking up. Since that's a very straightforward gene, these are most likely real. We have some further down, some CYP2D6, and it actually says a *4 on line 14. But again, we have to handle that very carefully to make sure that we're looking at the real gene, and we haven't determined that yet, is how much interference are we getting from the pseudogene. So we really liked this approach, and potential, even with our exome test, is to be able to give a pharmacogenetic result back, not based on the entire exome, not based on even 50 known genes, but based on the known genes with the known variants that we can interpret. So in going on then, genome sequencing is very expensive. Exome sequencing is also expensive. So what if we wanted just a test for pharmacogenetics, but have a very comprehensive test? So we've done a gene panel with a haloplex enrichment and running an Illumina. This does increase the depth of coverage, so the problem that we had with the CYP4F2 example we wouldn't have that type of a problem because we would have a greater depth of coverage. These would include all of our single gene tests, and then things that there are guidelines for, and things recognized as being clinically relevant. Now we recognize that we're not going to be able to pick up everything by our gene panel, but this gives us an opportunity to supplement by PCR. So we can do a long-range PCR to actually avoid the pseudogene and look only at the real gene. We can look then for translocations or gene conversions. We can do this by long-range PCR. This is how we're doing our CYP2D6 gene anyway in the clinical laboratory. There's been some initial work to see if we can simply include it with our haloplex enrichment so that we're not doing a second sequencing run. That looks promising. We need to do more work on that. But the exciting thing about doing this type of a gene panel is that we can then package it into specialties. And we don't need to look even at all of these 50 genes for every patient. So we could package it into things such as pain management, psychiatry, cardiology, or even oncology, looking at the germline variants. This will greatly help our interpretation if we do narrow it down to simply the genes involved with pain management. But the exciting part here is truthfully being able to then have a single test in the genetics laboratory, but being able to really focus on the clinical issue at hand, such as pain management and or one of the other specialties. Now I just wanted to bring up problematic CYP2D6 again, and this is still, we have to be very cautious. As I mentioned before, the pseudogene 2D7, deletions, duplications of functional as well as non-functional alleles. But we may be able to pick up the duplications of non-functional alleles by next generation sequencing. This is an area that I would really like to get into the data more. There are known gene conversions that we need to design specifically for them. The one question about the pseudogenes is, does the alignment include it? We need to be very cautious in the interpretation for those genes that we know have pseudogenes. But we could, the probes will hybridize to the pseudogenes, but you may be able to get the greater specificity by the alignment. But we are planning to fill in by long-range PCR. There's another issue that is difficult with CYP2D6. It, like others, are using the star allele nomenclatures, but the traditional nomenclature is based on the reference M3388. And so there are now, and that actually, one of the reference genes actually does not include one of the exons, so an exon is deleted in it. So if you start numbering at the first of the gene, you can go to a certain position, such as 600 in this. But then after that, you need to add a different number to come up with the standard nomenclature. So we need to translate all of the star alleles and all of the traditional nomenclature into standard nomenclature that will work with the next generation sequencing. And again, we still won't be doing the haplotyping, even though there may be some chance in the future that we could do more molecular haplotyping. But again, 100C to T is a star 10, whether as we have a 100C to T plus the 1846, it will be the star 4. So far the informatics has not been developed for next generation sequencing to take these into account. So we have some informatics work to do as well. So here is a gene panel variant review that I quickly pulled up and tried to look at overall. And I did pick up a CYP2D6 variant. In this case it was called 622 del A, Well, the other numbering is 775 del A, and if you look at this, this does become star 3. So my next step is to go and see if this is real. And a part of that is the validation of this for a clinical setting of taking known samples that we know the genotype to, or taking these samples and regenotyping them with our single gene assays, or by saying our sequencing. This just shows the alignment, and if you can look at in there, you can see approximately half of the reads have the deletion of this A. So the alignments actually look pretty good with this. Again, we need to then confirm. I wanted to use then the CYP2C19 target to show that there are ways, just some of the ways we need to carefully go through gene by gene. So we use the Integrative Genomics Viewer, IGV, to review coverage, and that's actually what I showed you on the previous slide. We review the full gene and the targeted variants to make sure that we have coverage. If we want to do the full gene, we need to have the coverage of the entire gene. If we only want the targeted variants, we can look at only that. So the table here is our attempt to translate the known star alleles into chromosome positions. So it has the RS number as well as the star nomenclature, the standard nomenclature, C dot numbering, and then the exon it's in, and then the chromosome position to look specifically for it. And so we have been working to go through all of the known variants and get them translated into chromosome positions. So I wanted to show you, here's our CYP2C9 in the IGV, and we pull up our reference gene and we can look specifically within that region. Not only that, we can then go into the coverage areas that we can see specifically that variant and look to see what the coverage is. So this is certainly a way that we can need to very carefully and cautiously go through the each gene, each variant, and make sure that we have the coverage. And I just wanted to show you something where we don't have coverage. So we did attempt to put in and analyze around other variants that are not in the coding region, and the star 17 for 2C9 was not. It's in a 5' region of the gene, and when we looked at it, it is not covered. Now with that said, when we looked at our IL-28B regions that we tried to design specifically for the single nucleotide variants and others, then yes, we had designed that, we had captured it. So this is an area that we will now need to go back and revisit to see if we can then be able to include the star 17, since that is a clinically important variant for us. So, as I said in the beginning, we're just stepping into this. There's a lot of very complex data by our genomic sequencing, but it can be done. So our pharmacogenetics data can be done from the exomes and from the gene panels. It obviously can be done from genomes as well, but I didn't have genomes to work with. I worked with the exomes. There are major biological and analytical challenges with segmental duplications, translocations, and even with simply changing to a standard nomenclature. I actually did some guidelines for the American College of Medical Genetics with CYP2D6 nomenclature, and we did put an appendix to that for common variants in 2D6 where we did the translation for you. And then the clinical interpretation, since that is so critical for clinical laboratories, that we do need to be able to do variants that we know how to evaluate. And so even for our exome, we would then focus on either our known PGx genes, or initially we'll go to our PGx variants and use the PGx genes then as more of a research type project until we can get a better understanding. And I want to acknowledge an awfully lot of people who have helped with this in our group. In specific, Dr. Mao and Dr. Bayrak-Toitemir have been instrumental in getting the next generation sequencing into our laboratory. Dr. McMillan is our pharmacologist that I work closely with in pharmacogenetics, and Dr. Milas is our research scientist in pharmacogenetics that has done much of this work, if not all of this work. And I appreciate the help that she is giving us and the understanding we're getting. This is an exciting area and a very fun project to look at for clinical laboratories. But we do need to have be careful in what we are doing, because this is clinical results that we would be giving. And I want to then mention the CME code that's available. And that concludes my presentation, and we have a few minutes for questions. And I see some questions have come in. And the first question is there any disadvantage in whole genome sequencing? There are advantages and disadvantages. The advantages obviously are that you're getting the entire genome, and so if you can't find something in your first analysis, you can then look into the deep intronic regions, or you can look in intergenic regions. Some of the disadvantages is that it still doesn't give a very high depth of coverage, so we won't be able to have 100, 200 types reads, and so we may be getting more false positives or even false negatives with a whole genome sequencing. Oh, I'm sorry, I was reading this is whole genome sequencing. This is whole gene sequencing. Let me finish with the whole genome sequencing, and then I'll go into this. So the other disadvantage for the whole genome sequencing is that it's still much more expensive than if we do the gene sequencing, a gene panel or even an exome. And one of the reasons is that we can include more samples at a time with our gene panels, so that we can really reduce the costs. Now, if the question really was the disadvantage in the gene sequencing, I still think gene sequencing, and even by Sanger sequencing, is still very important for genes that you know that they have the clinical presentation. So for example, in hemophilia, the factor VIII gene, people are typically diagnosed with hemophilia by factor VIII levels, and if they are low levels or low activity, then that's the diagnosis. Now, it doesn't make sense then for us to sequence the genome or an exome or even a gene panel when we know it's factor VIII. So I would imagine that in the near future, until costs become extremely competitive, that we will still be doing factor VIII by a Sanger gene sequencing. And there's a number of those examples. We are also using Sanger to confirm variants, so to make sure that we're not reporting out false positives. The second question is asking about long-range PCR. Let me see if I can. With long-range PCR, do you not fear to be exposed to PCR errors with no way to recognize them? And then long reads or paired end reads may be a safer approach to look at structural variants. That is true. The PCR errors, again, with our pharmacogenetic ones, most of these we have ways to look at them anyway. We use long-range PCR and we validated them. The PCR errors do exist, and I have seen that at a couple of times in my experience. But typically, in repeating reproducibility, we really haven't had that much problem with the errors of the PCR. Long reads is certainly a way to look at structural variants. Paired end reads, it may be a safer approach, but I'm still not sure that the reads are long enough to be able to handle it. So if we need to do the entire CYP2C19 gene to completely avoid the pseudogene, I'm not sure the next generation long reads are still going to be able to do this. I'm not sure I answered that question or not, but I tried to. And then the last question is open access database like PharmGKB up to date and accurate? I hope it is. I'm really not sure. I know that there are some updates and or some professional societies that we rely on to try to keep us updated, but that's one of the challenges we always will have. Let's see, we still have a couple more minutes. So I just got a little message that paired end reads with 454 will give you ranges up to 20 kilobases. So that's actually a very good one. Thank you for that information. The other question is, when do you see labs will be mandated to report incidental findings of whole genome sequencing? Right now, it's just a guideline. But given that guideline, given that it's from a professional society, I think most laboratories will be moving towards doing that. Most clinical labs. So even though we may not be mandated, that is, I think, where the field is going. Most of the times, there are very few mandates that we have, unless it gets into, potentially a CAP checklist where it becomes a requirement. But I haven't seen that happening. Whether it's mandated or not, I think the labs will be doing it. There are still questions and some disagreements with the ACMG, basically to say do it on everybody and not to basically ensure that they have given informed consent for it. There is a question about whether this is a large enough panel or should we add more genes, etc., to it. So there are still a lot of discussions to be had and a lot more work that will be happening. We have not started reporting this out in the clinical laboratory, but we are doing the groundwork to try to be able to see what we can be able to do. And with that, I don't see any more questions. And we are about 2 minutes before the hour. So I will thank everybody. Oh, one more question came in. Can I do it in 1 minute? Is it more useful to analyze the DNA sequence or the amino acid sequence of a protein structure to determine the cause of a disease? It's easier to analyze the DNA sequence than to determine the amino acid sequence. It is easier. DNA is more stable than proteins, especially if you need to look at enzymes. But the bottom line is whatever we do with the DNA sequence, we look to determine what it does to the protein. So it really is not as much the DNA sequence that is that determines, well, I shouldn't say that. The DNA sequence can determine the protein structure, but we always remember that what we are doing is trying to predict what would be happening to the protein as opposed to a DNA sequence. Okay, now that is, our time is up. Thank you very much for your attendance. Thank you for your questions. I hope I've been able to answer them at least somewhat for you. And we will end this seminar. Remind you about the CME codes if you need CME credit. Thank you very much.